Explain using the models, models and examples, the four principles of evolutionary theory by Darwin, evolution theory of natural selection by Darwin. So it means there are four principles. Okay. So first of all, we need to know what are these four principles. So once you know the principles, it will be very easy for you to answer any question that is related to this part, principles. Okay, so principle number one. So first of all, you should know, okay, let's write them. Principles. Okay, so the first principle, there should be variation between the organisms. Number two, there should be heritability. Heritability mean the organisms should be able to pass on the genes to the next generation. Number three is overproduction. Always it's said that the organisms produce more than survive or than the food availability or the resource availability. And the last one is reproductive success or reproductive advantage. Okay. Now, which of the following are amongst the conditions that are necessary for natural selection to occur? Number one, inheritable adaptations vary among individuals. So this is variation. So this is true. More individuals are born. This is true also than survive. This is overproduction. The environmental conditions are stable. No, the environmental conditions always change. So which one of the following are? So it means one and two. So one and two only. Keeping in mind all those four, we can just write them here. So number one, what's happening over here? It's variation. So organisms are a lot and they have different features. Number two, they are producing more organisms than survive. So you can see that the organisms are coming out of the area. So that's why we say more organisms are produced than they survive. Heritability is like when they have the reproductive advantage, leading to reproductive advantage. Heritability. So, and they have the characters which help them to be safe. So these are all camouflaging over here, and they will be safe, or rather than the others which are highlighted. Blue, green will be picked up easily. Now, in the last part, this is reproductive advantage. So all those who survive, they will reproduce and they will adapt and their number will increase. And you can see now the red is a lot. So they are increasing a lot. Define the term evolution. It's quite straightforward. Rate, okay, change in species, or you can say population over time. So if you say organism, it will go wrong. Remember that you have to say change in species or population. So this is very important. While if you say organism, it will go wrong. Differentiate between evolution, we just said over here, and natural selection. So natural selection is basically a mechanism that leads to evolution. So basically this is a mechanism, this is a part of the process. Okay, next, explain how natural selection is different. For natural selection, basically by nature, and here we consider environment as a nature. Okay, and artificial selection is by human, by scientists, by farmers, by breeders, anything that is related to man or human, that will be considered as the artificial selection. So which of the following example can be used to illustrate the artificial selection but not natural selection? So it means there are three natural selections. Okay, so let's check. Moth, dark colored moth become more common, so it is natural selection because it is happening naturally. Wolves evolve thicker. Okay, this is natural selection. Certain bacterial resistance, this is natural selection. Farmers, here comes the word that we are looking for. Farmers, so it means, or the breeders, see? So this is 100% answer for artificial selection. Differentiate between derived traits and ancestral traits. That's what we have to see now. Derived traits are like unique to a species. Okay, derived traits are unique to a species, while ancestral traits are common between species. Okay, so this is how the keywords you can make. For example, backbone is common between fish, snake, frog, bird, and mammal. On the other hand, the birds, they have feather, and mammal, they have hair. They are unique to them, so that's why these are the derived traits. So feathers and the hair, feather and hair are derived traits. Okay, derived traits are inherited from common ancestors. No, they are not inherited. Ancestral traits are found in both the common ancestor and its descendants, while derived traits are unique. Okay, these are the keywords. See, and ancestors and descendants. So between species. So this is the correct answer. Derived traits are shared by all species. No, they are unique. Ancestral traits develop after species diverge. No, that's the derived trait that diverge. Okay. Which of the following is an ancestral trait in character in mammal? As I told you, ancestral. So we do mammals normally don't have tail, and even if they have the tail, that's like not really an ancestral character that we focus. For example, fish and mammal also have some common ancestral traits. So here is a derived trait for mammals. Derived trait for mammals. Feathers is a derived trait for birds, while the backbone is the correct answer. Which of the following is a derived trait for mammal? As I told you already here. Explain evidences of evolution from morphology. What is morphology? Morphology is the study of shape. So when you study the shape of the organisms, how they look like, this is called as morphology. And here we have homologous structures, vestigial structures, analogous structures, and comparative embryology when you compare the embryo of different organisms. Description of each structure. Homologous structure. So how you define homologous structures? um, Structures which have so basically homologous structures, you can say similar structures which have different functions. So these are called as the homologous. For example, the four limbs of human, bat, and horse, let's say. While on the other hand, analogous structures are different structures. They are different structures with similar functions. So for example, wing of a bat and insect, both help them to fly, but the structure is different. The last one is the vestigial structure. So here we can say the structures that are the reduced form of functional structures in the ancestors. Okay, functional structures in ancestors or in other species. For example, wisdom tooth in human. There is also more like appendix. Appendix is another example. Tailbone is another example. In the table below, write the letter of the description of the correctly matches with each listed. Okay, so we need to read the description and match it with the headings. Okay, remains, whenever you have remains, it's fossil, so it's A. The reduced form, we just said over here, you can see up here, reduced form, so it is vestigial structure, so it's B. I'm not reading all. See, when you learn the keywords, it's easy for you. Here, the early and the pre-birth stages, it means they are talking about the embryo. So this is, no, sorry, it's here. Common ancestry based on the metabolic molecules like proteins or enzymes or amino acids. So when we talk about these, sometime also DNA they take the nucleotide. So this is comparative molecular biology. When you see the DNA and all that, like this one, okay, like this one. So this is comparative molecular biology. This is comparative molecular evidence, and this is embryo. So of course, we will not say embryo because we need to know the evidence. So evidence is comparative embryology. Here it's like a rock, so it's remains of the organisms, it's fossil evidence. You have the two organs in number two which are similar in structure, so of course we can't see what is their function, so that's why we will say them as homologous structures. And the last one over here, they are trying to show you the reduced form of an organ in a fish, a whale, that's called as the vestigial structures. Okay, moving to next. Explain the evidence of evolution from comparative molecular biology. As we said, we DNA, protein, amino acid, anything that is discussed that will be the related to comparative molecular biology. And if you talk about the distribution of the living things, that's geographic distribution, and that's also known as biogeography. So for example, earth is geo, living things is bio, and distribution is graphy. So now you can make it biogeography. So that's the answer over here. Moving to the next one. Which of the following demonstrates how geographic distribution provides evidence for evolution? So, of course, geographic distribution tells us about the unique organisms where they exist and how they are separate from other. So, answer is B. Describe the types of the adaptations in living organisms. Okay, here also you need to learn the keywords, and these keywords are very, very important to understand. Okay, for example, if you are talking about the camouflage, okay, so you need to look for a word that means blend. It blends with the background, it's like that. Okay, so when you are talking about mimicry, you need to look for copy. So either the copy is keyword, or you can look for look like, look alike, or look like. Antimicrobial resistance, it means you will have something related to disease, something related to bacteria, or maybe they say survive by antibiotic. Okay, so this is how these are the keywords that you look for. Which of the following correctly describes the camouflage in polar bears? What I told you, what you need to look for the camouflage blend. Okay, so we will look for the blend. Ability to smell, no. Digging holes, no. Fast swimming in cold water, no. Having fur that can blend. See, so this is the keyword, and that's the answer. Which of the following, write the letter example in the table below, correctly matches each listed type of adaptations? So as I told you above, camouflage, you need to look for the blend. Okay, let's try to look for blend. Here is the blend. Okay, so the answer for the camouflage is B. I told you mimicry, you need to look for copy or look like. So here you can see the look like. Okay, so the harmless scarlets have walled appearance look like. So the answer is C. And of course the remaining, if you don't know, even this is A. But here you can have bacteria infection, So like if you have bacteria or infection, something I told, you can go for the bacterial resistance. Next, summarize the 5 conditions of Hardy-Weinberg equilibrium principle. So first of all, we need to know the 5 conditions. Okay, so condition number 1. Sorry. Large population. Condition number 2, when there is a large population, they have variation. And based on this variation, basically variation will not make a point over here. So let's erase it. Variation will not make a point. We need to know large population, they have random mating. Number 3, no natural selection. Number 4, no mutation. And number 5, that's no migration. And you can see migration or no gene flow. Migration also have 2 parts, immigration and emigration. So no immigration, no emigration as well. Okay, a large population. So here we need to look for question. A large population of salamander living in certain ecosystem, and individuals of the population are mating randomly. So 2 conditions are mentioned already over here. This is number 1, this is number 2. List 3 others. So we need to list 3 more. Okay, we need to list 3 more. So you can easily pick 3 more from up. So you will go for no natural selection, no mutation, no migration. Now we come to the calculations of the Hardy-Weinberg related questions. Okay, so you have apply the allelic frequency within the population using the Hardy-Weinberg equilibrium equation. One equation is given to you here, while another equation is P plus Q is equal to 1. Okay, now the most important thing over here, you need to know what do these mean. Okay, that's the most important. Let me write them here. P plus Q is equal to 1, while P square plus 2PQ plus Q square is equal to 1. This is another. So P is dominant allele. It's the frequency of dominant allele, while Q is the frequency of the recessive allele. P square, since it is a dominant allele, but now it is dominant genotype, so it is homozygous dominant. Okay, so this is the genotype frequency. It's like this P dot P, or you can say P square. Q square is homozygous recessive, same thing. So you have Q dot Q, or you have Q square, while on the other hand you can see up 2PQ. P and Q, they come together, they make heterozygous. So this is heterozygous. So whenever you have to find out of all these 5, so we listed 5, right? So whenever you have to find these 5, like if you want to find the dominant allele, you want to find the recessive allele, if you want to find the homozygous dominant, heterozygous, or homozygous recessive, so you can easily use part of this equation to go with that. Okay, let's do some practice questions. In a population, the dominant allele frequency P is 0.20. Okay, just quickly write it down. P is 0.20. Which of the following is the heterozygous dominant? And underline it and say heterozygous dominant. Heterozygous is PQ, but don't forget the 2. So you need to find this one. This is question mark. You have P already. Now you want to find Q. So you will use this equation. P plus Q is equal to 1. So you can easily say Q is equal to 1 minus P, that is 1 minus 0.2. It's going to be 0.8. Now you can put it into this equation, 2 into 0.2 into 0.8. So that's going to be your answer. 0.32. So 0.32 is your answer for this question. Now, if in case they use the same question like p is zero point 0.2 and they want you to find homozygous dominant, so you will just take the square of the homozygous dominant. Okay, so for example, if you want to in the same question, I'm talking about the same question. Okay, so if you want to find homozygous homozygous dominant, so how you will find homozygous dominant? You have already dominant allele, that's 0 0.2, so p is 0 0.2. Now we want to make it homozygous, it means p square. So if you take the square of one side, you need to take the square of the other side, so answer will be 0 0.04. Okay, if they want you to find homozygous recessive, so you find q as you did already. So you got q is equal to 0 0.8, you do the q square and you take the square of the other side, and that will be going to be 0 0.64. So see, like, all kind of options are available over here. And if they just want you to ask the in a population, the dominant allele frequency is this. Okay, and they want you to find recessive allele frequency, recessive allele frequency. So it means that they want you to find what? They want you to find q. So just q, and you find it q here, so that will be the answer. So in one question you have all the options to do the questions. Keeping in mind what we did up, we will apply it same now in question 32. In a population of rodents, the frequency of dominant allele. Okay, now they didn't write P with it. Okay, be very careful. So we are going to write P is equal to 0 0.4. Which of the following represents the frequency of the homozygous recessive? Look, as I told you, they can ask you anything. So go up and see what we need. Homozygous recessive q square, right? We want to find this. To find q square, the first thing you need to find q. So 1 p plus q is equal to 1. Q is equal to 1 minus p, that is 1 minus 0 0.4, so it is 0 0.6. And q square is equal to 0 0.6 square, 0 0.36, that's the answer. Now you can also find the homozygous dominant, you can also find heterozygous in the question. So if you want to find heterozygous for this question, so it is 2 into p is 0 0.4, q is 0 0.6, so 0 0.48 is the answer. If you want to find p square, okay, p square, so that's so easy, 0 0.4 square, and that will be 0 0.16. So this is how everything works over here. Describe using examples the genetic drift. Now, what is genetic drift? Genetic drift is a random change in allele frequency. Okay, so it's random change in allele frequency, and normally this is a decrease in the frequency, like this change is a decrease, and this is taking place in small population. There are two types of the genetic drift, two mechanisms: founder effect and bottleneck effect. Founder effect is when part of the organism, part of population move to new place. Okay, move to new place. But over here in the bottleneck, they decrease to lowest and then they increase again. Okay, a small group of frog became isolated from their original population to a small island, which resulted in decrease. As I told you, it's a decrease in the upper part. I told you allele frequency decrease. So now I can easily say that this is genetic drift. Which of the four, when a small population settles into a new location? Look, I told you into a new place. So new place, I can simply go for bottleneck effect. I didn't want to waste more time once I know the keywords. A small population of cheetah is experiencing a sharp decline. As I told you here, there is a decrease to lowest allelic frequency, and separate from is due to habitat loss. So of course, this is bottleneck effect. Explain how mutation. What is a mutation now? Random change in DNA. And of course, whenever there is a random change in DNA, it is either increasing the allele frequency or decreasing the allele frequency. Okay, so which of the following best explains how mutations affect? Mutations only? No. Only? No. Mutation always? No. Mutation may increase or decrease? Yes. Okay. Explain how natural selection influences fitness and phenotypic variation, stabilizing selection, directional selection, and disruptive selection of the organism, and sexual selection as well. The graph below illustrates a type of selection that is called which type of selection. So here the most important thing, you need to look for the original population and what's happening over here after selection. So you see, like, there are three parts you need to focus: low extreme, upper extreme, and the average. So what's happening here, the extremes are ending and the average is being selected. So this is stabilizing selection. So when the average is selected, this is stabilizing. In this diagram, it's moving in one direction. So you can see the direction of the arrow, it's going in one direction, so it is directional. These are the three graphs that I can easily check and discuss with you. Focus on original and the new in all, original and new, original and new. So here the extremes are not selected. They are, sorry, 1 minute. So here it's moving it towards the one extreme, you can say. So it's called as directional. In this one, the extremes are not selected, only the average one is selected. So this is stabilizing. And here the average is not selected while the extremes are selected. So this is disruptive. Okay. Explain how reproductive isolation prevents the gene flow and drives speciation, like the prezygotic and postzygotic. That's what we have to say. So here we need to focus mainly on how this is happening, the reproductive isolation. So reproductive isolation is either pre or post. What do we mean by pre? Pre mean before zygote formation, and post mean after zygote formation. They result in isolating mechanisms and result in speciation. Speciation is what? Process in which new species are formed. In which new species are formed. And there are two type of the speciation, and they are: number one, allopatric, and number two, sorry, allopatric and sympatric. We will discuss them. Complete the table to define how prezygotic and postzygotic isolate mechanisms and to show how they contribute to reproductive isolation. So definitions I already told you up. Prezygotic. What do we mean by prezygotic? It occurs before zygote formation, while postzygotic it occurs after zygote formation. Descriptions. So we need to say description like how this is happening. So of course there is prezygotic. So there there must be a barrier that prevents reproduction. For example, the barrier can be time, the barrier can be place, the barrier can be habitat. Okay, so there are many different types of the barriers. While on the other hand, in the post-zygotic, we know that the zygote is formed, but the hybrid is sterile. The hybrid is sterile, or maybe it doesn't reach to the the adult stage. How does it contribute to reproductive isolation? So this one, it keeps the genetic materials isolated, keeps the genetic materials isolated, while the second one, it prevents from development. Now, compare and contrast the allopathic A for away. They go away from each other. It means that it involves a geographical barrier. Okay, it involves geographical barrier, while S for the sympatric in the same area, so they get isolated in the same area. So when you are having a geographical barrier as shown in the image below, so there is possibly a river that's passing between them. So identify the type of the speciation. So this is allopathic. Okay, allopathic, and that is occurring in the mice in the population. And explain your answer. And how you can explain your answer? I told you, you can simply say that there is a geographical barrier. So the two the two population the two population were formed after a geographical barrier. Okay, next. Distinguish between the two theories regarding, so what are the two theories? Gradualism and punctuated equilibrium. So we need to know what is gradualism and what is punctuated equilibrium. Gradualism is something which is slow and regular intervals of events, okay, for the evolution. While punctuated one is long stability periods during the evolution periods, or you can say long gaps. So gradualism suggests evolution occurs in rapid bursts. No, gradualism is slow, so this is wrong. Gradualism describes a slow, continuous process, yes. While punctuated equilibrium suggests long periods, yes, that's the correct one. Both gradualism and punctuated equilibrium describe evolutionary changes at the same rate. Same rate, no. Punctuated equilibrium occurs in small populations, while gradualism applies to large, only to large, no. With that, we reach to end of the topic. Let's stop here and continue the second topic later.